for selecting this paper to present in this highlight talk. And as we just heard, we usually believe that DNA sequences are about the same in all over the cells. And the DNA sequences in every cell is exactly the same when you take inside a random mutation. And we also used to believe that the RNA sequence is identical copy of the DNA origin, where it is derived from. Letting aside splicing, just remove part of the DNA, but eventually the core sequence, the same nucleotides, stays the same. And if we know the DNA sequence, we can use the genetic table and eventually translate it into RNA and proteins, and we know what is all the genetic information flow. However, there are protein families that didn't follow this rule and can alter the DNA and the RNA sequences into other components. For example, one of the protein family is the other protein family. The other protein family modify A, adenosine, into I, inosine, at the RNA level. And I is read as a G. So we have a little more complex genetic table than we used to think of, because A can be translated eventually as a G. Another protein family is the ApoBec. The ApoBec dominate C into U, and they can work either on RNA or DNA. So we have a much more complex real genetic table. And I will just brief a very brief introduction for this enzyme protein family. So the A2 editing is taken, the enzyme that is responsible for A2 editing is the ADARs protein. In human, we have three ADARs, ADAR 1, 2, and 3. They dominate the target by the diamidase domain and bind to the target by the double-strand RNA binding domain. So usually editing takes place in double-strand structure. And this is how editing is typically look like. You see in the genome, a very clean sequence, while in the RNA, that derived from the same locus, you see clusters, typically clusters, of mismatches of A to G. This was a prediction, mathematical prediction of alignment of ESTs to the genome, and this is lab validation when we sequence DNA, which are very clean, and RNA from the same tissues corresponding person, and you can see here the traces of the G that actually is echoes of inosines. And the role of editing in this non-coding region, because editing takes place mostly in allo repeats in human, now start to be revealing, and there are several mechanisms that control the RNA regulation via binding to inosines. The other protein family is the ApoBec. ApoBec can dominate RNA, for example, ApoBec1 dominate ApoB, and can dominate DNA, and the most famous member of this family is AID. These proteins dominate the locuses of the hemoglobin, and in large, we can say that is responsible for the light diversity in the immune system. But there are several other members of this protein family, and most of them dominate DNA and believe to dominate mainly evasion of viral sequences. And this is how it works. The retrovirus, when it's introduced to the cell, the ApoBec protein dominate the cDNA that emerge from the reverse transcription of this DNA, and dominate some of the C into U in the minus strand. So you eventually will see in the plus strand mismatches of the type G2A. And in order to eliminate this defense mechanism, HIV, for example, have specific protein, VIF, that is sole purpose is to put ubiquitin on the ApoBec3G and send it to the proteasome. So this is very powerful mechanism. And a few years ago, the Schwartz group demonstrated very nicely that ApoBec can also dominate endogenous retroviruses by introducing endogenous retro element into a cell with ApoBec3G. And here again, you can see like cluster of mismatches. Almost all of them are G2A, derived from ApoBec. So we decided to look computationally to find editing events. And the source of sequence of trace that we decided to use was the trace archive. Short RNA-seq, DNA-seq from the current technology are too short to find mismatches that are occurring in large clusters. And Trace Archive contains the raw material, genetic material that was used to, for example, build the genomic assembly of many organisms. And when we took this data, it contained about 2 billion traces. It didn't increase much more recently. And we select 10 organisms, and we align all the traces to the reference genome, hopefully to find evidence for DNA editing if it's DNA, or RNA editing if it's cDNA. And in order to be very careful in the alignment and not to have a false positive, we have a very stringent alignment criteria. So we took very long alignment, at least 400 base per long, at least 97 identity, and this means that if the cluster is too big, we'll miss it. And we didn't allow even one single insertion or deletion. And if there was any doubt about the genomic location, we didn't took it. So we aligned this procedure for 10 genome, and we start receiving clusters that look actually very nice, like a result of DNA editing. For example, here you can see trace. When you align to the genome, you see a cluster of G2A, and more nicely, we believe it was more nicely, it all have the same sequence motif around the mismatches, which all was G2A. And we see more and more and more such traces. So at the beginning, we were very, very happy. But it feels strange because we found that all the traces we found was only G2A. So when we look for a clustering with larger and larger number of mismatches, it became clear that most of them are G2A, some of the T2G. But when we look for traces with the same motif around these mismatches, there is a clear overrepresentation only of G2A. And this is very strange because the sequence derived from both strands. So this didn't look as a result of biological meaning, but more as an artifact. And indeed, this is very common artifact in the sequencing artifact we found. So this is genomic places, and 2 traces aligned to this position. You can see a very clean sequence here. But here you can see a little more noisy sequence. And this sequence, although the consensus is okay, you can see there is some noise. And the most typical noise is a lagging. You can see here, for example, that the noise always lag in one's base pairs from the chromatograms in the next. This red belongs to this red. This black belongs to this black. So we don't really care about it, but if you look here in the clean chromatogram, you see very interesting feature of sequencing that when you have G between 2A, and actually before A and G, the G peak is very, very low. And this is, if the sequence is clean, you don't have any problem with it. But if you combine this feature, that low G between 2A, and the lagging noise, you find something like this, that the lagging became dominant, and the noise lead for a false identification of this base coding. So this is a reason for the big majority of the sequences, mismatches we found. So in order to remove these mismatches and sequencing error, we focus only on very high quality scores. Actually, when you look for quality score above 40, which is 99.99% quality, you see that, as expected, you find mostly translation mutation of all type, and transition mutation, not translation. However, this sequencing error actually entered for several genomic databases. For example, for DB-SNP and HapMap, because determination of SNP is based on majority vote of the trace in specific position. And we found that in our set, and this is probably a huge underestimate of number of this sequencing error, we found 12,000 events that take place in HapMap, and we look at HapMap, almost all of them are homozygotic for the G, and there is no G2A because the platform used in HapMap is a chip and not a sequencing. While it's relatively very rare to see that all persons in all the population are homozygote for this position. So we believe there are large number of SNP derived from this sequencing error. And actually not only in SNP, but also in the genomic consensus, we probably affected by this artifact, because, for example, in Drosophila, we see no evidence for mismatches. And the reason is not because the quality score, and we check it in all genomic sequences, centers. It's not only because the quality was much better, it's just because the coverage was much lower. So the mismatches become part of the consensus. So after knowing that, we focused on very high quality score, mismatches, and we of course now have a very small set of widths, but here we start seeing candidates for DNA editing. And as expected, they enrich in the regions that are considered to be reasonably retrotransposon, like ALY, the most new member of ALY, the recently most active. And all these mismatches are extremely high quality. And again, it's enriched in regions that believe to have a retrotransposition. And in addition, in the trace archives, there are cDNA traces. This allows us to find editing events, RNA editing events. And RNA editing should be A to G, but because sequences are derived from the same both strand, we anticipate to see if it's RNA editing both A to G and T to C. And indeed, when we look in the trace archive of cDNA, we see no enrichment of A to G and T to C. When we look for the DNA trace, we didn't see again this enrichment. When we look in the high quality mismatches of RNA, you can see that almost all the widths, all the mismatches derive from A to G and complementary to C, suggesting this is real editing sites. And we know, for example, that editing, the other don't like to edit after the letter G. G upstream to the editing site. And here again, you can see that G is depleted both in human Xenophus before the editing sites only in the high quality set, but not in the not enriched set. Moreover, we know that in human, editing takes place mainly in ALU repeats, as again, indeed, most of these sites located inside ALU repeats. Interestingly, we found large number of editing sites in Xenophus, and Xenophus till now actually have, as far as I know, almost no documented editing events. Also, the other enzyme was cloned at first in Xenophus. So now we have a lot of sites that can be tested and learned for editing. This is an example of editing events in Xenophus. This position have several ESTs that support this region. And you can see the clear cluster of A to G mismatches. And as I told you, other bind to double strand structure, and here you can see the double strand structure formed by this region. And the editing take place as expected in position that obey to the sequencing motif of ADAR. So I want to end up with that I believe, I hope, I convince you that quality score is very important, and errors in the sequencing enter probably to the genome reference and dbSNP and several other resources. But DNA and RNA editing event can be detected in the trace archive data, and recently, and I didn't show here, we developed several more methods to find DNA editing events and find a large number of such events in retrotransposome. And we introduced in this approach several candidates for somatic/germline DNA editing events. And we found large number of RNA editing events in human and specifically in Xenophus. And I want to thank the people that contribute to this work, mainly to Sasha, and Tomer, Tom, and Shai that helped tremendously with this work. Thank you very much.